Welcome back to my YouTube channel Medical Classes by Dr. Srinidhi Kumar Acharya. Please don't forget to subscribe my channel and also please give your valuable comments. So we were discussing about fetal growth and as well as development. So in that context, we have already completed the formation of the trilaminar layer, how the ectoderm, endoderm and mesoderm has been formed. Now today we will have the continuation of the same. Now if you see the cross-sectional view of this particular three layers, okay, so then it can be seen as shown in the figure. Ectoderm is nothing but the uppermost part, and below that there will be mesoderm, and in between there will be the and in between then it is the notochord, and then below is the endoderm. So first you have ectoderm, then you have got a mesoderm, and in between there will be a notochord, and below that there will be endoderm. So this is the arrangement. Now the notochord touches the endoderm, and that portion will go into disintegrate. In the further development, what happens is the notochord, the area where the notochord touches the endoderm, that particular portion will undergo disintegration, and notochord will proliferate over the edges of the cut portion of the endoderm. So notochord start proliferating over the edges of the cut portion of the endoderm. So in between there will be mesoderm. We know that in between there will be mesoderm as shown. Now this particular area where notochord proliferate over the edges of the cut portion of the endoderm, so that particular portion is called as notochordal plate. Okay, so notochordal plate, so which it will fill the place of neuroenteric canal as it disappears. So that will going to fill the place of the neuroenteric canal. Now here the things are very clear again. Okay, so where is the neural tube, then the notochord, then comes mesoderm, ectoderm, endoderm, etc. The dorsal portion and as well as a ventral portion. Okay, so you can see over here in the cross-sectional view. Now this is again amniotic cavity. You can see then mesoderm where it is, ectoderm, then comes endoderm, then the amniotic cavity is there, and this particular cavity, blastocyst cavity is there. So primitive streak, where actually the primitive streak in the beginning itself, in the beginning where the actually changes start in the epiblast. Okay, so that area primitive streak will be there. So everything is very clear in this picture. Now what happens later? Later the notochord becomes the solid mass, and it separates from the endoderm, and it is disconnected, and the disconnected portion of the endoderm again fills. Okay, so later the notochord becomes the solid mass and separate from the endoderm, and the disconnected portion will be again filled back by the filled back. Okay, so now it looks like a solid rod like structure, and now it is called as definitive notochord. So in the picture you can see now it is called as definitive notochord. Later it play a very vital role in the development of the nervous system. Why it is so important is this particular definitive notochord. So this plays a very important role or vital role in the development of nervous system and other systems of the body. Notochordal plate towards the ectoderm. Okay, notochordal plate which is present towards the ectoderm proliferate, and the cells of the ectoderm to form the neural plate, and while the cells of the mesoderm beside the proliferate to form the gastrointestinal system. So this is again what is important. Notochordal plate towards the ectoderm proliferate the cells of the notochordal plate which is present towards the ectoderm. The cells will undergo the proliferation to form the neural plate. That neural plate becomes in future plays a very important role in the formation of the nervous system, while cells of the mesoderm besides they proliferate to form the gastrointestinal system. Everything has been given in the figure, so it is proliferate to form the gastrointestinal system. So eventually all the three layers are derived from the ectoderm. One thing is very clear, eventually all the three layers what we are talking, so they are basically derived from the ectoderm, isn't it? In the previous class also we have seen it is the epiblast, the changes start in the epiblast. So then same cells undergo the proliferation, and then it forms the endoderm. Then the same cells they undergo to form the mesoderm. Lastly, the epiblast is called as ectoderm. So ultimately, everything is derived from the ectoderm. So the ectoderm become converted into skin in future. Okay, when the fetus fully grown, the ectoderm becomes converted into skin. The mesoderm get converted into muscle and urogenital system. Notochord provides the central axis for the development. Notochord, it provides the central axis for the development, and endoderm development helps in the gastrointestinal system. Gastrointestinal system, so that will be developed from the endoderm. So body has outside with the ectodermic derivatives, with the mesodermal and then innermost endodermal derivatives. So this is what is actually explained in our science, Ayurveda, where this is Matraja Bhava, Pitraja Bhava. Okay, Pitraja Bhavas are the outermost portions, okay, ectodermal origin. All the ectodermal origin, the skeletal growth, skin growth, then CNS growth. Actually, these all things are derived from the ectoderm actually. Okay, then innermost structures of the body, maybe viscera, etc. So they are derived from the endodermal derivatives. So in between, so there will be mesodermal derivatives. Now from the upper part and the lower part of the cylindrical trilaminar layer, from the upper and lower part of the cylindrical trilaminar layer, 4 buds will come out. Okay, so this is the next part. So from this, now the 3-layer formation is over. From this 3-layer formation, then it folds, just like what you say masala dosa, when it is folded, so it gets folded, so it becomes cylindrical. It becomes cylindrical. So now upper end and lower end is formed. Okay, now from this, actually in the beginning, the 4 buds will come out. 4 buds will come out, and there will be only ecto and mesodermal will go into it. No ectoderm or organs. Okay, so in these 4 buds, what we say, okay, in these 4 buds, the structures, these 4 buds, they contain only ectodermal and mesodermal layer. The endodermal layer will not come in these 4 buds. The 4 buds which come out, in that it contains only the ectoderm and as well as a mesoderm. The endoderm, the innermost layer, so it will not come, because endoderm is related to the formation of the organs and all. Okay, so it will not come. So this has been very clearly told in Ayurveda that pratame maase kalali jayate dvitiye panchapindika bhavati. So when they say dvitiya maase ganam sanjayate, and in the tritiya maase panchapindika bhavati. So they says that the 4 buds will come out. 4 buds will come out from this particular ectoderm, this particular trilaminar layer. Now this is about the formation of the trilaminar layer. In the next class, we will see the clinical correlations of this formation of the trilaminar layer, which is very important clinically. Thank you very much. We will continue in the next class. Till then, goodbye.